Alright, today I'm going to take you through glycolysis, or anaerobic glycolysis. This will just be a very generic summary of what's going on. I've drawn some of the structures, but they're not perfect. There are a couple of things that are not right in terms of structure, so if you're trying to figure out what the molecules look like, this is not what you want to see. Refer to a biochemistry book for that. This will just highlight some of the major concepts with glycolysis during exercise in a skeletal muscle. So you start off with glucose. Glucose floating around in the plasma can be absorbed into the blood. Once it gets absorbed into the muscle cell, once it's into the muscle cell, you go through a process to make glucose 6-phosphate, where you put a phosphate on the glucose. It requires ATP, so you break down ATP in one of these first steps and form ADP and hydrogen. The consequence of that hydrogen forming is acidity, and that hydrogen ion can leak out of the muscle cell and can lower the pH in the muscle or the area surrounding the muscle. Once you have the glucose 6-phosphate formed, if you're not exercising, your body usually will go ahead and store it in the form of glycogen, where it just hooks up a bunch of glucose molecules into a big pinwheel or web-like structure, saving it for later. At the onset of exercise, you'll usually use the glycogen as your main source of glucose. It'll be broken down again into glucose 6-phosphate, this time not requiring ATP. You've already made that investment. You don't have to make it again. So if you use glycogen, you actually use one less ATP than if you use glucose during exercise. Small little difference there. So you have the glucose 6-phosphate. That and then goes through a couple of steps where it requires another ATP to phosphorylate the molecule again, to phosphorylate the sugar, forming fructose 1,6-bisphosphate. The drawing of this molecule isn't entirely accurate. It usually is a pentose. It has like a pentagon shape. Here I've drawn it as still looking like a hexagon, just for convenience and clarity. But if you want to look at the shapes, go look at a biochem book. In the process of phosphorylating that, you use another ATP, forming ADP and another hydrogen ion, which again may dissociate out of the muscle cell, contributing to the acidity of exercise. Once you have the fructose 1,6-bisphosphate, it's broken down by phosphofructokinase, PFK. This is one of the major regulating enzymes, rate-limiting enzymes of glycolysis. It breaks the sugar molecule into two factors. You have 2 3-carbon molecules, which are then processed a little bit more, and in the next step, an NADH molecule is produced. You take NAD and produce an NADH, which can then later be used for aerobic metabolism if you have enough mitochondria present. If you don't have the mitochondria present, that NADH can actually become a liability. Then you go through another step where now you're reaping the benefits of all the investments you made up earlier. Now you get ATP out. So since we've broken it into 2 molecules, we're actually getting 2 ATPs for this next step. We get energy release as this molecule is processed a little more. You go through a couple more steps and more ATP is released. So now if you look at it, we've actually released 4 ATP, and we had to invest 2 ATP up in the beginning. So if you're using glucose during exercise, your glycolysis will give you a net of 2 ATP because you have to spend 2 ATP and then you get 4 ATP in the end. If you're starting just from glycogen, you've already invested that ATP a couple hours ago, not during exercise, so you don't have to worry about that, and your net will actually be 3 ATP because you only invest 1 ATP during the exercise and get 4 ATP back. So it's a nice little deal you get there. The end product of this is pyruvate, a 3-carbon molecule. Now pyruvate can have a couple of destinies. One, if you have mitochondria available in the cell, that pyruvate can be processed and participate in aerobic metabolism. Another thing with the mitochondria, if you have mitochondria present, the NADH that you produce in this step earlier, right after phosphofructokinase, when you get that NADH produced, that NADH can migrate down to the mitochondria and participate in aerobic metabolism and actually help you generate more ATP. And then that NAD is freed up to go participate back in the glycolysis earlier. And so this is a nice little loop if you have the mitochondria available, where you can get the NADH formed, get ATP for that NADH in mitochondrial respiration, and then you have the NAD available to participate in glycolysis. Another perk of having the mitochondria around is that the hydrogen ions that you form during ATP hydrolysis that contribute to the acidity, those hydrogen ions can actually participate in mitochondrial respiration, and so if you have the mitochondria there, they actually tend to buffer how much hydrogen ions are floating around in the cell, and that you have less increase in acidity. In contrast, if you don't have those mitochondria floating around, we'll look at it over on this side of the diagram, on the left side of the diagram, you have to do something else with the NADH. So this NADH, you can take it down to the pyruvate, and the pyruvate will accept it and form lactate. And that lactate leaks out of the cell. And now, so you've gotten that NADH back to an NAD, which now can go back up to participate in glycolysis earlier on in the process. Forming the lactate allows glycolysis to keep on going by making the NAD available to facilitate glycolysis. In the process, you form lactate, which goes out of the cell and then can be consumed by other cells that have lots of mitochondria, like the heart. The liver also seems to consume the lactate. There's a couple other things. Neighboring cells with mitochondria can consume that lactate. If you notice, there's a bidirectional arrow here, so the lactate can actually be converted back up to the pyruvate and go over to the mitochondria, and that's what I'm illustrating there. That it doesn't always have to stay as lactate, but you can actually get something out of the lactate in terms of energy production. So this has been just a guide to what happens during glycolysis in a muscle cell during exercise. One more key point that I want to highlight is that the lactate is not really the source of the acidity that occurs during exercise. That acidity seems to really be coming from the hydrolysis of ATP and a couple of other things. But lactate or lactic acid itself, it doesn't really seem to be around. It's always in the salt form lactate. So technically it's incorrect to say that lactic acid is being observed during exercise, or it's incorrect to blame lactic acid for any kind of fatigue. The acids or hydrogen ions are coming further upstream in glycolysis before you ever even create lactate. So again, this is a summary of glycolysis during exercise, skeletal muscle exercise. Hopefully you find it useful, and that's all I've got.